symposium. And now I invite Professor Shankar Ramakrishnan from our department to chair this session on digital medicine. And so Shankar was the previous head of the BSV department, and he has been one of the is very actively involved in the MFCEM initiative from the very beginning. And he has also known our keynote speaker, Professor Shankar, for a long time. So there is a personal connection there as well. So without further ado, I invite Shankar to come on stage and introduce the speaker.
Thank you, Nitin. So I would like to welcome all of you for the first session of MFCEM inaugural symposium. And the first speaker of digital medicine session is very aptly Professor Shankar Subramaniam. And for most of us, he is not new. We know him. We know about Shankar. And his connection to IIT Kanpur goes back to several decades. And I am very happy to introduce him. And Professor Shankar Subramaniam is a distinguished professor of bioengineering, bioinformatics, and systems biology, computer science and engineering, cellular and molecular medicine, and nanoengineering at UCSD. Professor Subramaniam has played a key role in raising national awareness for training and research in bioinformatics. He served as a member of the National Institute for Health NIH Directors Advisory Committee on Bioinformatics. In 2008, he was awarded the Faculty Excellence in Research Award at the University of California at San Diego. In 2011, he was appointed as a distinguished scientist at San Diego Supercomputing Center. Dr. Subramaniam has served on the External Advisory Board for several bio and biomedical engineering departments, including Johns Hopkins, Case Western Reserve, UPenn, Georgia Tech, Rice University, and University of Texas, Austin. Dr. Subramaniam has also served on Bioinformatics and Biotechnology Advisory Council for Virginia Tech, the University of Illinois at Chicago, and on the Scientific Advisory Board of several biotech and bioinformatics companies. He is currently an overseas advisor for the Department of Biotechnology of the Government of India and a member of European Science Foundation panel. So with this introduction, I would like to invite Professor Shankar Subramaniam to deliver his lecture.
Thanks. Thank you, Shankar. Good morning, everyone. I'm sure you're sick of me by now. I've been occupying this podium for a long time, yesterday and this morning. I promise you, after this talk, I will vanish from the podium, at least. And so, welcome, everyone. And so this morning, I wanted to tell you about ongoing work in my lab for the last several years. And our interest was, I'll tell you in a little bit of story-like line, how our interest was kindled in this area and why we do this research. And I'll give you a brief overview of this. So this is the goal of my talk here, to tell you about how is the brain reprogrammed in Alzheimer's disease and how we could really delve into this using digital medicine. By digital medicine, I mean large volumes of data measured by multiple modalities. So I'm going to tell you about that in a few minutes. So like Dr. Gong Bao did yesterday, I have funding from many, many sources. I have 9 NIH grants and foundation grants and all that, which makes this possible for me to do the kinds of work. We work in multiple areas of research in systems biology and systems medicine, but I'm going to only talk today about neuropathologies, and this is focused entirely on Alzheimer's disease today. And so what is Alzheimer's disease? I mean, this is something that most of us, if you live long enough, we are going to get dementia at some level or the other. We'll lose some, brain cells. We keep losing brain cells. Eventually, some of us will end up, or some people end up in getting Alzheimer's disease, which is a specific type of extended dementia. I'll tell you all about it in a minute. And the hallmark of Alzheimer's disease is that you have shrunken brain size, dying neuron with tangles, plaques, formation of big plaques in the brain. And this is kind of the hallmark of Alzheimer's disease. The time people detect it is they start losing memory, but most of us don't admit that we are losing memory. Even if I go to, I'm sure losing a lot of things, I forget things, but I'm not going to go regularly and check myself to see if I have Alzheimer's disease. So by the time people find out that they have AD, it's too late. It's by the time, and the way they find out is they start forgetting things more and more and more. You go to do cognitive tests, and the cognitive tests lead to some kind of imaging. And when you do imaging, you find your plaques and tangles, and that's when you realize that you have Alzheimer's disease. Okay, that's one modality. And so I'm going to tell you a little bit of story. Let me start with the story. So this is a kind of a map of the world, and I put dots randomly. It looks like I put dots randomly in different places. What makes these dots related to each other, and what is the meaning of this? So I'm going to give you a small story about this. I'm going to focus on one dot, which is actually in Italy. And in fact, if I zoom in on this region, I'm going to get this region, a province called Catanzaro. And when you look at the Google map of Catanzaro, what you're going to see is this map of a place. This is Catanzaro, the red highlighted region. And there's a place called Girifalco, a small town. I think this is not working anymore. And in case you want to fix it, I can just use my hand. So the one highlight of this place is you got to see. I didn't write this down. This is in the map. There's something called Ospedale Psichiatrico in the Girifalco, which is a psychiatric hospital of Girifalco. What's so special about this hospital? So it turns out that this hospital started in 1640. In the year 1640, it was not a hospital. It was started by a priest. Because there are a group of people who were demented and who were called crazy, mad, psychotic, all those things. So they kept the records of all these people. So this is a set of families, a few families. Okay, thank you so much. So they just kept a record of these families. And so it turns out that this record has continued for a long time. And what's the relationship with this record? So if you go to, now the slide thing is not working, but that's okay. Is there a way I can move it here in any other way? Sorry about that, folks. So I'll tell you the story while he's pulling it up. So it turns out that now turn the clock forward to 1904. In 1904, I think this is not connected to that. This thingy here.
Yeah, connected.
It's connected now. It's not moving. Just change the battery in this, that's all. If you change the battery in the other one. Okay. So let's, I'm sorry. We're having technical issues, but don't worry about it. I'll tell you the story and then you'll see the slide after that. So in 1904, there's a person whose name, who was found, whose records, these records are maintained for a long time. But it turns out that this person's record was indicative of a mutation in a given gene. And this one mutation in the given gene, and for this I want to show you this because it's a really exciting story. And it was published actually more recently. I'll give Nitin a chance to. Thank you, Nitin. I think this should work. Okay. I'm often technologically challenged, but this is even beyond me. Okay. Fantastic. So this is the picture of the hospital in Girifalco. And you can see that this is very ancient. In fact, this record is the record of Angela R. And you can see this record is 1905. You can see the date here. And this record tells you about this person's afflictions. It just describes the afflictions of this person. So turn the clock back to 2014. They had a sample of this individual. They dug up a sample of this individual. They could sequence this individual. right? And they found out that the sequence showed that she had a familial case of Alzheimer's disease, which means that she had a mutation in one of the genes which showed that she had Alzheimer's disease. And Alois Alzheimer, in the same time frame, 1904, was the first one to recognize that there is a set of diseases of the brain associated with neurodegeneration, which is called after him as Alzheimer's disease. Now let's turn back the clock. Let's look at the history. If you trace back the origin of this individual's family, you can see that everything in dark is affected patients. So if the gene was derived from the parent, the person was affected with Alzheimer's disease. Okay, the gene was derived from the patient. So in fact, this family, which is why this region in this place, the whole family, the Girifalco Hospital has a record from 1640 until today of the family members who are affected by diseases. They were called by different names and different things and so forth. Now let's go back to the map. It turns out that there are people in Melbourne, Australia, and Buenos Aires, Argentina, these red dots. These members had exactly the same mutation in the same gene. And when they traced back where they originated from, they didn't know that they were of Italian origin from Catanzaro. But when they traced back the origin, they found out that they migrated, their ancestors had migrated. Without new genomes, whole genome sequencing, they were able to find out that they were related. And that's what relates all these dots. For example, patients here, 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 and here, they have the same type of mutation. Different from the Angela R mutation. These people here have the same mutation and so forth. So these are all mutations in just 3 genes. In fact, I'm going to show you those 3 genes. Those, maybe I didn't get them here. I have it here. These genes, which are all coming from presenilin 1, presenilin 2, or APP. These are the 3 genes. If there are mutations, specific mutations in those genes, it's a question of not if you are going to get Alzheimer's disease. It's a question of when you are going to get Alzheimer's disease. They are 100% penetrant disease genes. Okay, so now let's go forward a little bit. What's the problem with AD? AD is the sixth leading cause of death in the US. 5.8 million Americans are living with AD. 1 in 10 people of age 65 and older have AD. By 2020, AD and related dementia will cost $305 billion. By 2050, it's $1.1 trillion. That's the cost for Alzheimer's disease. Older African Americans are twice as likely to have AD than older Caucasians. After investments of tens of billions of dollars, NIH has invested, several tens of billions of dollars in public money. There's not a single cure for Alzheimer's disease. Okay? There's absolutely nothing. So the disease I told you about is called familial Alzheimer's disease. Turns out that people who are old, like 75, 80, and 85, also get Alzheimer's disease without this single mutations, right? Which means there must be a genetic background in these people, which is causing the Alzheimer's disease and dementia and Alzheimer's disease. Correct? In fact, if you look at the Indian population now. In fact, I was talking to somebody who works on neurodegeneration from Bangalore, from NIMHANS in Bangalore. They said in Indian population, they don't even recognize, because people didn't live that long to know that they had, AD. And now it's starting to show that people have dementia and AD. So it turns out that sporadic AD, which is what it goes by, is actually more common. It's greater than 65 years. And familial AD, which I told you about, is the one that is less than 65. In fact, I'll show you the age group. The age group is, I should have it in the next slide. The age group is 45 years, 58 years, 47 years, 51 years. That's kind of the plus or minus age group. And most of these people die within 10 years after they have AD. So this is really the familial Alzheimer's disease. And the mutations I told you about are in these 3 genes, in presenilin 1, which is this simple protein here, presenilin 2, or APP, which is a substrate for these proteins. And these are the mutations mapped in red. Anytime you have that kind of a mutation, and now we can all do 23andMe genome sequencing, and we can figure out, hey, do we have that mutation? In which case, what can we do about changing our lifestyle? So this is really the hallmark of this. Now, the question that often is asked, first question that was asked was, why is this gene causing, why is the mutation in this gene causing Alzheimer's disease? Let's try to answer that question. So this is what I told you about previously. So let me go back to the brain of AD. This is healthy brain. This is Alzheimer's brain. It's got plaques and tangles. What are these plaques? These plaques are nothing but misfolded proteins, which are just simply having these peptides, which are processed by the protein I told you about. That processing gives rise to aberrant processing. And when you have an aberrant processing, they aggregate. And these aggregations, oligomers, they form these aggregates. These are amyloid plaques, and they call fibrils, amyloid fibrils. They are 100 nanometers. They're thicker and thicker. They grow in the brain. They kill the cells, and eventually you lose your memory and so forth. This was a thought that was going around till about 20 years ago, till 15 years ago, or 5 years ago, when people thought that amyloids were the cause of the death. So since 1960, the amount of money invested in amyloid research, amyloid-based drug research, is billions of dollars in public funding and multiples of billions of tens of billions of dollars, hundreds of billions of dollars by pharmaceutical industry, right? Let's see what happens. So I'm going to tell you the story from Eli Lilly, which is a pharmaceutical company that is, and Eli Lilly invested $3 billion in AD therapeutics. This is from Indiana Business Journal. And you can see that they invested $3 billion in AD therapeutics. Okay. They did a phase 3 trial of a drug called semaglutide for treatment of Alzheimer's disease. And this trial went on for 20 years till 2015. Then they showed in New York Times, an article appeared in I don't remember exactly, 2020, let me see what it is. Here it is. I can't read very well from there. 2010. It was shown that Lilly stops Alzheimer's disease drug trials. And they published this in New England Journal of Medicine. The trial said the placebo, as compared to placebo, semaglutide did not improve the cognitive status, and patients receiving higher dose had a worsening of functional ability. However, I want to tell you the plaques were gone. In fact, when they did imaging, there were no plaques, right? But they didn't recover memory. So the question is, why is it that you dissolve plaques, you get rid of them, but you don't get back your memory? What's the problem? So now there are other companies. For example, there are 6 companies: Roche, Johnson & Johnson, Pfizer, Biogen, Genentech, Lilly. All of them did drugs which are based on plaques, and all of them failed. And in fact, there's an article in Nature Reviews that Alzheimer's failure raises questions about modifying strategies. And the ultimate journal, Forbes, said amyloid hypothesis is dead. So, which really means that you don't assume that amyloids are going to cause the thing. So this kind of prompted us. So why are these drugs a failure? So this is the reason I'm going to tell you, for students who are here, you have to always have a passion or a question that you ask, and then you try to address that question in the ways that you can think about. So the question was, why do these drugs fail? Why are Alzheimer's drug discoveries a failure? So the first thing you should know is we don't have good model systems. If you have diabetes, if you have liver disease, your mouse, good models, they're great models, right? If you have brain disorders, Alzheimer's disease, mice don't get Alzheimer's disease. Monkeys, primates don't get Alzheimer's disease. We get Alzheimer's disease. So therefore, this is really a big issue, right? There are no good models, and we cannot interrogatively investigate the human brain. There's no way to do it. So therefore, there's no good model systems. That's the first issue. Second issue, we attacked the wrong mechanisms, removing plaques. In fact, I had an intuition years ago that the reason why all these drugs are a failure is because it's too late in the game by the time you get plaques. And we said, why is it the case? So the mechanisms are wrong. So our goal was to find out what are the mechanisms which will address the disease. The third question is, even when you do find a target, let's say you find a target and you have a drug, how do you screen? You cannot give the drug to a human being, and FDA will never let you inject the drug into your brain to see if you're going to survive or you're going to do better. So there's no good way of screening. So our goal was, we said, okay, we want to bring engineering approaches towards these problems. This is the goal. I was telling you yesterday that engineering is really coming into focus in terms of human pathologies. So make personalized brain organoids. So we said, okay, what if I do the following? what if I take my brain and make an avatar of my brain? Can I do it? The answer is yes. Today we can do an avatar of our brain to some extent and see how it behaves. So that's the number one question. Second is we want to develop a drug reactor screening. We want to have a high throughput robotic automated way of screening. So these avatars, we can screen them for all the drugs and show that they work. That's a good starting point. Then FDA will say, okay, we'll give you approval for doing clinical trials. Third, we want to have quantitative assessments and we want to target the right mechanisms. So the next half hour, I'm going to walk you through all this story of how we came through with this process. I'll go through some slides very fast because it's more technology than anything else, but some slides I'll walk through slowly to give you the idea. So here's the way we started thinking about it. We said, okay, what if I take my skin and get stem cells from my skin, convert the stem cells into neurons? There'll be more my neurons or your neuron because it'll be individualized and personalized, right? I'll have the same genetic background. So I make these neurons. Let me see how different are these neurons from the neuron of patients who have an Alzheimer's mutation. Okay, the question is very simple. We take mutations, we take neurons from people who have these familial mutations, we take a non-demented control patient neuron, we compare the two to see are these neurons similar, how do they evolve and so forth. That's the first question. That's a model system. Now the question will come about, okay, it's great that you're creating a stem cell derived neuron, but what about the brain is more complex than single cells, so therefore you need to have something more. So that, okay, we can make organoids out of these things, which means we can take these neurons, build them into complex organoids that contain neurons, astrocytes, microglia, oligodendrocytes, the entire cell population of the brain, make it look like a mini brain, and then now we can look at the disease in more order. So this is our solution, engineering solution, and I'll walk you through how we implement this and all those things. Second, we want to ask the question, if I take these neurons, can I find out how they are different? This is where data comes into picture. We do millions of different experiments to give you millions of pieces of data, which tell you how these two neurons are different. We do transcriptomic sequencing, which means we want to look at how they are different at the expression of genes. We want to look at epigenetics. We want to look at how the chromatin is folded in these neurons. I want to look at what mechanisms are implicated in the Alzheimer's brain. Okay, so this was the quest of what we set out to do. And then we could build a reactor-like system here where we can take the library of compounds. We can then screen this library of compounds against these neurons or against the organoids. So we can really do a high throughput way of saying, hey, what drugs work? What combinations of drugs work? I'll give you an example of this as we go along. So this is our goal. So the first paper we published was a long time ago in 2020 in our dictionary. So we showed that the reason why all the Alzheimer's drugs based on plaques failed was because they attacked the wrong mechanism. It's way too late. The first things that happen is that the neurons undergo de-differentiation. In fact, Gang was referring to it yesterday in some of his studies as well. So what do you mean by de-differentiation? That means the neurons, they are no longer neurons. They lose their neuronness. As a consequence, they lose synaptic connections. We are who we are because of our brain's ability to connect and fire. And so if you lose our synaptic connections, then we lose memory. That makes sense, right? So therefore we lose memory. And then it goes through a whole series of processes. So our first goal was to find out what are the mechanisms that are different between a normal, non-demented control human being. In fact, we took extreme case of an 85-year-old person who was completely cognitively normal to make it as contrasting as possible. Then we took these Alzheimer's patient samples and then we transformed them into neurons and looked at what's different between the two. So the next 10 minutes, I'll tell you the story of this thing. So we looked at all these different mutations. They're all familial Alzheimer's patients who all have Alzheimer's disease. There are different families. In fact, one of these families is the family of Angela R., who had the defect from Girifalco's hospital from Catanzaro. So one of this was from that family patient. So we took all these different mutations and then we transformed these into neurons. And then we try to look at what is the effect of those. And I'm going to tell you first the story about all the presenilin-1 mutations. In fact, they are the most lethal mutations because they're the shortest lifespan, presenilin-1. Presenilin-2 is next and APP is third. First, I'll show you presenilin-1, then I'll walk you into presenilin-2 and APP. Okay, so our goal is very simple. We just take these neurons. Thanks to Yamanaka factors, we can transform them into human-induced pluripotent cells. And one of my colleagues developed a technology where you can transform them into neurons greater than 90% and glial cells. So we transformed all of these into neurons. Let's see what happens. So we use technology, crazy amounts of technology, RNA sequencing, chip sequencing, methylation sequencing, and we are still doing high-C, but we did ATAC sequencing. We did all these kinds of different sequencing, and you don't understand the magnitude or amount of data if I don't tell you. It's just enormous amounts of data. And so this is where the digital medicine comes into picture, where we are talking about. So this is our first idea to give you first a high-level view. All these combined approaches towards various sequencing approaches told us a simple story. It said, in the disease case, you have dedifferentiation, which means you're no longer a neuron, or neurons come from a germ layer called ectoderm. You're no longer ectodermal. You start behaving like you're a confused cell. And so therefore, you're no longer a neuronal cell. And so then you have cell cycle reentry. Once we form our brains are terminal, we reach terminal state of cell cycle. We don't cell cycle. Our brains don't reproduce themselves, right? So neuronal cells don't reproduce. However, this neuronal cells from Alzheimer's patients showed like as if they are going through a cell cycle. Okay, so then the third thing is there's neuron lineage repression. Neuron lineage, things that were canonically called neurons, their genes, they were downregulated. And synaptic function loss. This is the most important thing. We lose synapse like crazy, which means all memory is slowly going away in these patients. Okay. So then we said, okay, so I don't want to walk you through this slide. It's a very complex slide. So our goal was to look at all these different presenilin-1 patients. We asked the question, okay, if you did it in one cell, then somebody will come and tell you, hey, this is such a special case that you're looking at. You're not looking at the general case of what's going to happen in all these cells. So we said, okay, we'll look at 4 different presenilin-1 patients, and we'll look at what's common amongst all of them. And when we look at what's common amongst all of them, I just want to give you a high-level view. They'll become pluripotent, which means they're no longer neurons. They look more like stem cells than like neurons. And second, they go through cell cycle. They start cell cycle. They have inflammation. They have downregulating neuronal specification. That means anything that's neuron-like is downregulated. For example, glutaminergic receptors, GABA receptors, neuron-specific channels, they're all downregulated. And then you have dedifferentiation, which is upregulated, which is essentially you're going back into something. When I say dedifferentiation, what I mean is you're going back towards a reverse direction from being a neuron. Okay. I said, okay, all right. So what can we do about it if we find this? We find all these specific targets of cell cycle, pluripotency, dedifferentiation, neuron specification. So now we have a handle on what goes wrong in the brain. And it's not one thing that goes wrong. There are about half a dozen things that we can think of as going wrong. How do you fix it? I'll walk you through that in a few minutes. Before we do that, this is actually a more complex way of looking at it. Who regulates these changes? Who regulates these changes are these genes. They're all transcription factors or regulators. For example, everything in red, orange, yellow is increasing. Everything in green is decreasing. So these are the regulating or controlling factors. Can we control them? Can we use drugs to look at these things? Okay, I'll show you examples of how we can think about it. So cell cycle is an example, and I'm going to skip this slide mostly except to tell you, we know the precise mechanism by which these cells enter this cell cycle. Imagine this mutation here, presenilin-1. This is now essentially what it does is it takes a protein, breaks it down in the wrong way. As a consequence, it re-enters the cell cycle. And then it goes into cell cycle, but doesn't complete it, stops somewhere here. So we know precisely the mechanism of how the cell cycle is regulated, for example. So let me go to the next slide. So the next question then that came to us, you try to publish such things, they will tell you, you have to know why it happens. It's not sufficient to just show that it happens. Okay, why does it happen? So, okay, I had this idea. the cell state is changing, which means the cell is going away from being a neuron into a non-neuronal state. What does it mean? It means that the cell's directions, where it tells you what genes you express to be a cell, that is changing. And how do you know how to determine how that is changing? What decides the state of a cell is the chromatin state of the chromosomes, and it's folded and open and all that. Yesterday, I think I told you, if you take the entire chromosome 2 meters long, you compact it into a 100-micron nucleus, then you're really going to be compacted. So which means some parts are open, some parts are closed, and this is what decides whether you're a cardiomyocyte or whether you're a neuron or whether you're a skin cell and things like that. So we ask the question, how is the chromatin changing? There's a beautiful direct set of experiments you can do to look at chromatin landscape. And this set of experiments essentially is called chromatin accessibility ataxic measurements. You can look at how the chromatin shape changes by locally mapping, is it open or closed? Okay, it's a very simple question. Open region or closed region. If it is closed, it cannot express a gene. If it is open, it can express a gene. Now, if it turns out that in the neurons, you have genes, neuronal genes are expressed, that means that region must be open. If an Alzheimer's neuron, if you don't express those genes, that region must be closed. If the answer to this is yes, then we have a hypothesis that the chromatin determines the control of who is expressing and who is not expressing. Exactly what we see. In fact, forget the slide. So when we look at all the complex analysis of this, the analysis simply tells you exactly the same thing. In an Alzheimer's neuron, you lose chromatin accessibility for what's a neuron. You gain chromatin accessibility for what's a non-neuron. As a consequence, you express genes which are non-neuron-like. And you close regions which are synaptic genes, so you no longer have synaptic gene expression. As a consequence, you don't have synapses anymore. Okay, so that's what gives rise to dementia. So we now have a mechanism. To corroborate and to verify this, we actually looked at it. We looked at open and closed regions of chromatin through this experiment called ChIP-seq. That is, you can actually use a protein to pull down the regions that are open and closed. You look at the histones, and then you can say, hey, it's open or closed. When we did that, we got exactly the same result, which is essentially, we looked at the regions which are open and regions that are closed. And I'll show you that we precisely showed that there's a strong overlap between open and closed regions and the regions that are open and closed through another experiment. So we are corroborating that this is exactly what happens. Now we said, okay. So when we tried to publish this paper first in a high-profile journal, they said this is fantastic. However, you're doing it in a neuron derived from a stem cell. What about in the human brain? In the human brain, does the same thing happen? So we were so lucky that we got a post-mortem human brains which had mutations in exactly the same region, right? So we're able to do this experiment, and I'll show you in a minute. Are these same in vivo patient data? So we looked at patients. We looked at common genes between the post-mortem brain and the neuron samples. Guess what? When we did this experiment, we did exactly the same program. Look at the comparison, so stark here. Between, these are the mutations, this is the brain, M139T, this is the human brain sample. You can look at how similar pluripotency, dedifferentiation, cell cycle, neuronal specification are. The same things happen in the human brain, right? So therefore, this is really convincing that it is not just, we are just doing a cell experiment that is happening in the human brain. Okay, so then we showed, how do we explain the mechanism? So we published the paper where we showed that these regions in the normal brain, like all of your brains, are open or closed here, and these are open, so which is why your brain functions so beautifully, controlled by these samples. However, when you go to an Alzheimer's brain, guess what? Everything goes awry. All those regions that were originally open became closed. All those regions that were originally closed became open. And so as a consequence, your cell state is no longer a neuron, it is something else. Okay, now let me continue here. So we said this is our kind of a high-level view of what happens. You go from a stem cell into a normal brain. This is our terminal neuron. When we are children, this is what happens. In the Alzheimer's disease, you go back into a precursor-like cell, which kind of is no longer in no man's land or no person land, which means that you don't know whether you're a neuron or not a neuron. Okay, fantastic. So we said, okay. Then came the next question. What about mutations which are, we did presenilin 1. What about presenilin 2 and APP? They also give Alzheimer's disease, familial Alzheimer's disease. What about those mutations? I said, okay, we said, well, I'm going to walk very quickly through this. So we said, okay, we published this paper. This is in molecular psychiatry. And so it shows that systems level analysis shows exactly the same thing. We looked at common regions between all these different mutations and look at what happens. The end result, believe me when I tell you this, the end result is the same regions, the same things happen. There's a loss of neuronal specification. There is a gain in pluripotency, loss of synaptic connections. The same things happen. Okay, so we are convinced that this is published. We're convinced that this is really happening. So I'm going to skip these slides. It's all the complex sequences. But we did something slightly more interesting than that. So, okay, when you take all these mutations, some of these patients die earlier, some of these patients die later. Some are more severe, some are less severe. Can we predict this severity of disease from all this beautiful data that we are getting? The answer is yes. In fact, when you look at this data, we show neuronal function goes from here to here, decreases. Guess what? The lowest neuronal function are the ones which are the earliest diers, people who die most early. Then you got, and every single function that you look at, we have a pseudo trajectory or a pseudo map of saying which patient is going to benefit or which patient is going to die soon, which patient is going to die later. Okay, fantastic. So we have, this is our model. So we say that this is, let's see, yeah, this is your going into a synaptic regular neuron. In the case of the Alzheimer's disease, you have different stages. Some of them are less severe, more severe. They go into more precursor-like state. And so we now have a map of all these things where we can show exactly what happens to this brain. Let's go further. Then the question came. Then when we submitted all of this, I gave a talk in some big institution, and they said, hey, this is all great, but this is familial Alzheimer's disease. What about Alzheimer's disease of patients who are 70 years old or 80 years old and who get Alzheimer's disease? They don't have these mutations. And we don't know what mutations they have. So what about these patients? So we said, okay, we are very lucky that in UC San Diego, we have a Alzheimer's core from NIH. And we have a lot of patient samples, postmortem brain samples. We got 49 postmortem brain samples. And we took these brain samples in 3 different regions of the brain of these postmortem dead people samples. And then we analyzed these samples, the same technology using transcriptomic measurements. When we did that, we showed it was more complex, more heterogeneous, more confusing, or more things were happening. However, what was common was dedifferentiation, which means they were no longer neurons. They stopped behaving like neurons. We had increased inflammation and all that. So we published this in this journal called Molecular Brain. And we showed that exactly same things happen. I'm sorry, I don't see this. You show that neuronal function, lineage function is altered. Then there's altered lineage, and the chromatin pluripotency and inflammation increased. So this was the first time somebody had shown that there is similarity between the induced pluripotent stem cell derived data and human brain data, not just only in familial AD, but also in sporadic AD. So now I answered the first question. Can we build a model system? Yes. Can we find the molecular mechanisms associated with the model system? Yes. Can we now do drugs? So that's the next question, right? Therapeutics. So you said, okay, why did the Lilly drug fail? So the first question you ask is, why did the Lilly drug fail? I mean, it's after all supposed to went through 15 years of phase 2 clinical trials. Why did it fail? So we said, okay, let's do the following. Let's take these induced pluripotent stem cell derived neurons from familial Alzheimer's patients. Let's treat them with a Lilly drug. And for comparison, we'll treat them with another drug. Let's see whether the drug fixes the problems I saw in these neurons. If it fixes the problems, then it's a great drug, at least a starting point. If it doesn't fix, then we know why it failed. The answer is very simple. We did all kinds of complex experiments. I won't go into detail. How am I doing on time? I don't have a concept of time ever. Do I have another 20 minutes? 15 minutes, okay. So I'll just show you one thing. So therefore, when we did the experiment, first we did experiments in terms of titration of these things. We showed that if you use a Lilly drug, Lilly drug removes any ability, it removes all of the processed APP peptides, all the A beta 40, 38, 39, everything. So it really removes. So that's okay, except that we had another drug which removes only some of them, which is better. And so we said, okay, let's compare these drugs and see what happens. But nevertheless, the bottom line is that the response of the drugs targeting downregulate cell cycle. Remember I told you it goes back into cell cycle? These drugs do great in terms of regulating cell cycle. They remove plaques. They essentially, they don't have plaques anymore, so they remove plaques. But they don't do anything to the synapse. They don't restore your brain connectivity. Since they don't restore your brain connectivity, you're going to end up continuing to have dementia, right? We showed this elegantly. We showed this through a complex set of experiments using these gamma secretase drug targets. And we showed that the bottom line of all of this is very simple. I'm going to summarize this for you. We published this paper already in the journal called Alzheimer's and Dementia. And we showed that in the normal case, you're going into this, this is a normal case where you go from precursor into differentiated neuron. In Alzheimer's disease, you go into this state, in this bottom state, and you can go into intermediate stages. You can go into this stage with the Eli Lilly drug. You can go into this stage, but you'll never go back to this stage where you're completely restored the neurons. So therefore, we know that these mechanisms are not addressed by these drugs. Okay, next question. Can we address it by any other mechanisms to look at drugs? So we said, okay, we decided to look at the following. So let's take all the FDA-approved drugs. Let's take the drugs that are targeted against brain function, brain diseases, Parkinson's and all those diseases. So we are going to really do a workflow of drug pathway identification, and then we're going to take this and look at drug target identification. We are going to combine these things, and then we are going to look at what drugs can we try to look at these things. So when we look at this complex analysis, we showed that when you look at drugs that are involved in neurogenesis, drugs involved in synaptic plasticity, neurotransmitter receptor drugs, inflammation drugs, all these drugs, we take a combination of all these drugs and pathways, and then we try to build a complex machinery where we can look at all the predicted drugs, all the drugs we think might work. I mean, this is all computer stuff yet. It's not really, in experimental stuff. So we predict all this stuff. We look at, in fact, we look at predicted candidates found from chromatin accessibility. We find these are key drugs which we could try at. And right now we are screening all these drugs in singly and in combination so that we can go and look at what drugs work and what drugs don't work. So that's where we're at. Now comes an interesting question. We have Alzheimer's disease starts in some region in the brain. How does it spread? Is it the same in all the regions in the brain? Is it going to be unique to some regions? Can we fix drugs? If you have drugging, can we target it to specific regions of the brain, or is it going to be entire brain? So to address this question, we are very fortunate that we got, the original data generation was not in my lab for this. This was done already by somebody, but they just looked at it for some other reason. We got the data from 6 different regions of the brain. And in fact, we looked at entorhinal complex, hippocampus, mediotemporal gyrus, parietal complex, SFG, and visual cortex. We looked at all these different regions of the brain. And we were able to map the same thing, the transcriptomic map of these different regions of the brain to see which regions are more similar, more cause earlier effect and all that. And so when we did the comparison of each of these regions, we showed that in fact very similar mechanisms exist in many of them, but some more in others. For example, in the cortex regions, we had a greater effect of these mutations than in other regions. So we are now not only able to look at the mechanisms that are faulty in the brain, but we are able to localize them to different parts of the brain. In fact, now, as we speak, we are doing single cell sequencing in brain organoids, which can tell us exactly what regions of the brain initiate the process. We are not there yet. Okay, so I'm going to skip this slide. So now comes the next interesting question. These mechanisms are great in Alzheimer's disease. What about Parkinson's disease? What about Huntington's disease? What about ALS? Are there similar mechanisms? So I said, okay, we'll investigate them. So I had a very sharp student who said, okay, I'm going to really take on this job. I'm going to look at Parkinson's brain data. And in Parkinson's brain, many of you know that we have defects in substantia nigra, and we have, again, we have formation of plaques. Parkinson's disease brain has essentially formation of plaques due to specific set of proteins. Huntington's brain has similar things in terms of brain regions which are affected. That's the next slide. Huntington's brain has also defects in basal ganglia, which forms these repeats called CAG repeats. We said, okay, let's take these comparisons and see what happens in these brains. And this student of mine fits, he took all these different data sets from patient replicates from Huntington's disease, Parkinson's disease, ALS, and he did a comparative analysis of all these things. And guess what he found? Very simple. He found that the mechanisms are, in all these cases, Huntington's, this is Parkinson's, and then we have also ALS. All these cases, chromatin gets remodeled, just like we saw in Alzheimer's. Cell cycle reentry happens. Neuron specification goes down, and dedifferentiation goes up, and then immune response goes up. Essentially the same process, phenomenon, underlying phenomenon. So now comes the more interesting question. Why is this the case? So let me tell you, before I say why is this the case, let me tell you the story. So right now we are in the process of building organoids, cortical organoids, and we have a paper in Nature Protocols under final review. And I lost my slide here. For some reason, the slide doesn't show up here. So the bottom line is we now have 10 months of organoid growth in normal patient and Alzheimer's patient growth. So we can see the formation of different structures in this organoid growth. Okay, so now comes the interesting question. Why is it that so many different diseases show the same mechanism? And I'm going to give you my perspective on this and how we are approaching this problem. So in my lab, we have worked at so many different diseases of the liver, diseases of the muscle, diseases of vascular system. In all these cases, we went back and re-examined the results. In all these cases, what happens is that when you have a stress in a tissue or stress in a cell, the cell is reprogrammed or programmed to de-differentiate, to protect itself. Wound healing is an example of that. You cut yourself, the cell essentially goes into epithelial mesenchymal transition. Many of you know this, EMT. And the reason it goes into EMT is it protects itself and re-differentiates back into an epithelial skin cell, for example, or other cells, right? We know this from real experience. We found out this is to be true in the muscle, in the liver, in the vascular system. Anytime there's an injury, or anytime there is a stress, whether it is a toxic stress, oxidative stress, or any type of stress, the cell and tissue is reprogrammed so that it can go back into a protective state. It can build more fibrinogen and other proteins that form a moat around it. It can do inflammatory signals, which says that the cell is under danger. Those are 3 hallmarks of every single chronic disease. It happens in the brain too, except that in the brain, when it happens, you lose synaptic connections. So you're no longer able to go back into restoring your normal cortical structure. As a consequence, your brain gets impaired to a point that you don't have an ability to do this. So what's the next steps? So what we would like to do is to have an early marker or an early indicator, which is going to tell us that the cells are going to be reprogrammed. And if we can stop them, prevent them from being reprogrammed, then we have a chance of addressing dementia, addressing Alzheimer's disease, Parkinson's disease, Huntington's disease, or other brain disorders. This is a quest in my lab. How are we going about it? We are taking brain organoids from Alzheimer's patients. We are taking brain organoids from Parkinson's patients, Huntington's patients. We want to figure out how to restore or prevent dedifferentiation in these organoids from early stages. So we are monitoring them longitudinally across time, okay, going across time so that we can really understand how this happens. Now I'm going to finally tell you for the next 3 minutes or 4 minutes, I'm going to tell you a story which tells you how significant this is for other things. So let me walk you through that. So years ago, when I was really naive and stupid, and so I didn't really understand this concept. Now we understand this concept more clearly. At that time, we are talking about 10 years ago. We are doing research in collaboration with Inder Verma, who's well known, who's in Salk Institute. And we were working in this area called glioblastoma. I don't know how many of you know, glioblastoma is a cancer of the brain. It is a signature of death. When somebody gets a glioblastoma, it's not a matter of if they will survive. It's a question of whether it's days or weeks or months, a few months at the most. So Inder was working for a long time on glioblastoma, where he was looking at, all these different targets. He was interested in looking at drug targets, and so he came to us at one point and said that, hey, I'm really interested in looking at mouse models. We are going to induce glioma. We want to know whether this glioma that is induced in the mouse brain, are those tissue and cells, what are they like? If you tell us what they are like, we can try to understand the biology of what is happening to these cells. So he said, okay, great, we'll take up this challenge. Now, this brought a very interesting engineering challenge, and I'll tell you about it in just a minute. So this is our goal. Our goal was to really take these stem cells, take these neurons, and then figure out whether they are like stem cells or whether they are something else. So we are really trying to figure out. One problem that we had was when you induced glioma in the mouse brain, there are only a few cells. You don't have, it's not like putting a cell culture, induced pluripotent stem cell derived neuron where you have millions of cells you can deal with. Here you have thousands of cells. So we have so little mRNA that we can profile. We have little RNA we can profile. So we said, what is the solution around it? So I worked for, this was in 2012 or 13 or something. We worked very hard on trying to profile this. We went to Illumina, which is a big company in San Diego. So we told Illumina that, hey, we've got very little sample of this thing. Can you help us really profile this? They tried their best, and we could only get 30%, 40% mapping. So I said, okay, what's the engineering solution to this? So I came up with this idea that, okay, here's what we are going to do. The reason why we were not able to get more than 30% mapping was because of thermodynamics. It's just simple. We use the primers. The whole idea is they use primers to amplify the RNA. We said you can use primers to amplify. There are only some X number of primers, and these primers go and bind to the most obvious binding site. So they were amplifying only some nucleotides. They were not amplifying the entire mRNA. So we said, okay, how many primers do I need to cover the entire chromatin? That's a computational question, right? We said, how many primers do I need? So I said, okay, then I got somebody in my lab, a postdoc in my lab, Panko, said, why don't we go and find out how many primers do we need, minimum? And to our enormous surprise, we found that with 54 primers for human and mouse, we were able to cover the entire 99% of the transcriptome. So then we said, okay, let's design these primers. So I'm really not very sophisticated, so I said, let's design these primers. We designed the primers, and for the next 1 year, everything failed. Why did it fail? So then we found out that because these primers are small bits of nucleotides, they were falling off. They were not binding to the thing very well. So we said, okay, let's do the following. Let's do a low temperature primer design. Then we can do a high temperature design once we expand it. So this is called a design primer amplification strategy. Is a very bright student of mine, Vipul Bhargav. He worked for 2 years, his rear end off. At the end of the time, he was able to show that we were able to map 99%, I'm losing this, we were able to map, and we are able to show comparison quantitatively R squared of 0.94. Look at the mapping, we are able to map everything, 99% of the transcriptome. Now, this is, of course, a popular technology. Everybody in the world is using this technology. But this is the design primer technology that we developed in 2014 or something like that. Okay, then we showed validation of this technology. Now, I want to come back to the brain, glioblastoma. We applied it to the brain, and we said, these cells don't look like brain cells, the glioma cells. They look more like precursor cells. At that time, of course, I had no clue about dedifferentiation and all these things. So I just simply published a paper where we said, okay, these cells look much more like pluripotent cells. But we did more than that. We said, okay, let's look at what is the common element between this. We showed that the common things in the glioma were these three things: Wnt pathway, focal adhesion, and cell cycle. Now I can explain to you why this is the case, because cell cycle is upregulated, right? And pluripotency is upregulated, and the cells come together forming clumps. Now I know it, which is but then I didn't know it. So then, however, we found a common gene, one gene, this SPP1 common between these things. He said, so I told in there, let's knock this gene out in the mouse models and see if it removes cancer. Okay, if it does, then we have a proof of this hypothesis. That's exactly what we did. We knocked this one gene out. First, we did experiments with just simply using a knockdown of this. You can see this is cancer, knockdown, cancer goes away. Okay? We did the same thing with antibodies. We just said, okay, let's just do it with antibodies. This is all, cancer. Look at the antibody thing, it reduces. Okay? How about drugs? We said, okay, this is in astrocytes and neurons. We did in all kinds of cells. And we said, okay, let's look at what happens to the lifetime of these things. We looked at all these pathways, they're restored back. Looked at the lifetime of these things. We looked also experiments in the mouse. This is experiments in the mouse brain where we show that the cancer goes away. I'm going to skip this slide. The lifetime of the mouse. Look at the lifetime of the mouse. The mouse now started living longer and longer and longer by this knockdown. Very simple. This is just a brutal treatment on the mice. We take the mice and knock out the gene. It lives longer. Okay? Now, in fact, Inder is pursuing this where he's taking a small molecule drug target for glioblastoma. He is doing it as a clinical trial in India. He has a company in India, in Bangalore, and he's doing this in a clinical trial in Bangalore. Okay, so then what's the bottom line story? This is again the same story where we can show that this gene is really the gene that causes big death, and so just a story complete. So let me tell you the last slide. So what we showed was, we said, here's an example of what happens in a stem cell going into a neuron, astrocytes, oligodendrocyte. In the case of Alzheimer's, you're going back into a precursor-like state. Glioblastoma, you're going to a precursor state. In the Alzheimer's case, and in the Huntington's case, Parkinson's case, the cells don't reproduce. The cell cycle is not complete. In the cancer, the cell cycle is complete. So what happens? So you start producing more and more cancer. It grows, it proliferates. And this is true in not just brain cancer, it is true in all cancer. So we now have a central thinking or hypothesis. Under chronic stress, cells de-differentiate. Most of the times they recover back their native tissue state when the stress is relieved. Sometimes they don't recover back when the stress goes beyond a certain point, and they go into a cancer state. So we now have a kind of a hypothesis in terms of how to approach this problem. In Alzheimer's, we are going to approach this problem by doing targeted therapy, at first in the level of organoids. Then we are going to try to do it in a clinical trial at some level, hopefully. And as one of my philanthropy friends is very fond of saying, I was explaining to him this, how we are doing all this. He said, do something before you forget. So I want to stop with that. Thank you for listening to me. Oh, these are all the people involved.
Thank you so much, Shankar, for the very fascinating and amazing talk. So we have some time for a couple of quick questions. Ashwini.
Thank you, Subramaniam, for such a wonderful and enlightening talk, especially the system approach which you are utilizing. My question is basically, I was losing the track due to the fact that you have not talked about the role of aggregates in your organoid culture, and whether they are part of the toxicity or synaptic loss or not. So your thought on this, please.
Thank you. It's a very, very good question. So I want to divide your question into two parts. The first part is, when does the disease begin? And the second part is the plaque formation, once the plaques are formed, are they going to affect the brain? There are two distinct questions. Once the plaques build up to a certain stage, there's a physical crowding effect. There's an effect of really taking over cells, and cells are really, there's biomechanics, there's fluidics, which means there's no oxygen to these cells. So the cells are going to die and necrose, and this is how the death happens in Alzheimer's disease. But by the time we get to that point, can we look at, are they the origin? The answer now is very clear to the community. The origin of this is not the formation of aggregates. By the time aggregates are formed, we are already getting to a point where it's late. So the origin is much earlier. So our goal is to look at the origins. In the organoids, for example, brain organoids, we are starting to see the aggregation of these plaques. But in the 10-month time frame, we don't see the organoid dying yet. We do see diminution in the organoids and diminution in the neuronal synapse formation. But there's no, complete destruction of organoids. But maybe if you pursue it further, we are now only 10 months into building these organoids. We are going to extend up to 2 years if we can, and then we'll see what happens to the organoids. So the answer to your question is, it's a chicken and egg problem. Once you form the chicken, it can have all kinds of problems. That goes back to the issue, right? But our question is to catch it at the early stage to see what happens. Did I answer your question? Yeah.
You were also mentioning that probably aggregates are forming even later.
That's our current hypothesis. By the time aggregates, before that, there are many other processes initiated. So we want to prevent the initiation of these processes. Amitabh.
Shankar, so if one takeaway message from your presentation is that one, cells are de-differentiating, two, the chromatin accessibility is changing. So two questions coupled, related. That is, I was under the naive impression that cells that are more pluripotent, they would have higher chromatin accessibility because they might go to different kinds of lineages. And two is that if you look at really progenitor cells, do they have similar kind of chromatin architecture in terms of accessibility and not having it?
So they're both, you have a complex set of questions. They're all related and very, very good questions. So first conception that just proliferating cells have higher chromatin accessibility. They have higher chromatin accessibility for genes that are involved in cell cycle and proliferation. It doesn't mean that they have higher chromatin accessibility in all of the regions. That's number one. We know this from a large number of differentiation phenomena. So the answer to the one part of the question is, yes, there's increased chromatin accessibility for CDC, cell cycles, CD kinases, and so forth. However, I didn't show you the cell cycle diagram clearly. So there's reduced chromatin accessibility in, for example, neuronal specification and so forth. So it is not everything is open. It's a question of some things are open, some things are less open. If you take pluripotent stem cells are very interesting cells in the sense that they have the ability to form different germ layers. However, a lot of them are silenced unless they are induced to change in the direction. In our embryo, we have cues, extra embryonic cues, and these cues prime them to go from an ectoderm into a mesoderm into an endoderm. Till then, they are silent. They are happy to be just silent, yet being effective. Right? This is exactly what happens here. We are going into, in fact, when you have wound healing, epithelial, mesenchymal transition, mesenchymal cells are just that. They are able to preserve their ability to go back into epithelial cells, right? Otherwise, we wouldn't have formation of any tissue, many tissues. So that's the answer to the answer to it. So when we look at chromatin accessibility, the reason why I brought chromatin, cell state, this is a concept of cell state. In fact, our current work in breast cancer with some extremely exciting results about how these cell states change even within epithelial cells. I don't have time to tell you this. Maybe next time when I come to IIT Kanpur, I'll tell you. But this is really the question of what is a cell state? Can we determine it through chromatin topology, which tells you how to regulate things? Right? That's where we are at the moment.
Okay. Actually, there are so many, I can see hands. So just two more questions.
Keep it brief.
And then students and then.
Yeah. So I was just curious on your thoughts on what is the primary event that you think is responsible for Alzheimer's disease. Like you talked about the neuron-centric processes. What's the role of neuron-glia crosstalk in Alzheimer's disease pathogenesis?
Yeah. I think you ate enough chocolates today. You're really high energy. So this is a super question. This is absolutely the question that I get asked in many, many places. So it's all beautiful, the mechanism, how it happens, what type of drugging you're going to do. But what causes it? Very good question. So beautiful question. So in the case of familial Alzheimer's disease, we know the cause because it's a mutation in presenilin 1, presenilin 2, which are part of this enzyme complex, gamma secretase complex, which is the one that's responsible. So beautiful, clear story. What about in Alzheimer's, old patients who don't have these mutations? Our current hypothesis is stress is causing it. What is stress? It can be toxic stress. It can be oxidative stress. It can be, for example, there's not enough oxygen supply in that region of the brain because of the fact you have injury, brain injury, traumatic brain injury. In fact, I didn't tell you the story. We're now looking at traumatic brain injury, same phenomena. So you have essentially occlusion, no oxygen to these regions of the brain. That's stress. So we think stress in other tissues in human body, stress is the main cause. In fact, to do this thing, we published a very nice paper which showed that in endothelial cells, we stress the endothelial cell to see at what point the cell decides to die, at what point the cell recovers back into death. We call this as the nexus between life and death in a cell, right? So this is exactly what we think happens. We think we have a kind of a transition point at which point this tells the cell, hey, this is so much stress you cannot survive. Commit suicide, right? Otherwise it says, okay, we can recover from this stress. It recovers. So we looked at the suspension point between life and death in a cell. So we think stress is the cause. What stress? How do we originate? I don't know the answer to that. It's very complex, so we don't know the answer to that.
And a quick question. Yeah.
I think you should give it to Dhirendra. He has another question too after.
Shankar, really nice talk. I enjoyed it. Wanted to ask you two things. One was in the Eli Lilly trial, there was one statement at the end of the abstract which said that although it got rid of the plaques, but it made them prone to other diseases, including cancer. I don't know if your group was interested in looking at that aspect, but if you can throw some light on that.
So the reason I brought the cancer in the last part of my talk was exactly to address your question. So the reason why the Lilly drug, it did not completely address the cell cycle. It did not completely address the synaptic formation. So the first part of it is, even though it removed the plaques, it did not do that. Because it did not restore the cell back into a neuron, it's now a pluripotent cell. So what can it do? It starts reproducing.
Right.
When it starts reproducing, it is cancer.
True. The other thing was just the last slide during your conclusion. You said that your hypothesis now is that this is dedifferentiating into a slightly more progenitor type cell.
Correct.
Do you have a hold on the mechanisms that lead to that?
Yeah. In fact,
just wanted to complete that. And if you do, then you probably can also lead to, instead of therapy, you can also look at prevention, because preventive action can be taken for disallowing that progression towards.
So clearly, cell state change is the route which it takes. What does it mean? It means that the chromatin is going into a non-neuronal, non-specific cell state. Whether it is endothelial cell or neuron or whatever disease, it's going back into a precursor cell. How is it doing it? It's doing it through chromatin landscape changes. Now, but what is the cost? This is the question that she asked. The cost is that stress. So if you can alleviate that stress at some level, then we can probably restore. I don't know what the cause of the stress is in the brain. But in other cases, we know the causes of these things. For example, in liver disease, we looked at it in liver disease, going from non-alcoholic, from NAFLD, fatty liver disease, into steatohepatitis. We know that there is a well-defined set of factors that are controlling this. Diet, for example, alcohol, for example, or other things. So if you can alleviate these factors, can we reverse it back? And this is, other people are trying this now in going from NASH to NAFLD into reducing that state. So the answer to your question is yes. We know the type of mechanism, cell state change overall. Can we address it in some way? Now, there's a second way of thinking about it, and this is what we are really thinking about. When you are going to now go back into a precursor-like state, if I can have a factor which can keep it in the neuronal state or keep inducing it to go into a neuron, then it's a good strategy. In fact, one of the drugs I talked about, I didn't have a chance to go into detail, is exactly that, which tries to differentiate a stem cell into a neuron. And so this is what we're trying to do, to look at whether we can keep it in a neuronal state. And an experiment I didn't tell all of you about, which we're doing in collaboration with Cambridge, we're actually taking a normal stem cell and growing it into a neuron, monitoring it through the entire process of 70 different time points. So we can look at exactly how to trigger, at what stage can you trigger with what factor, the conversion to the next stage. And we are trying all those kinds of experiments now in organoids.
A quick question. Yep.
Yeah. So you have enormous amount of data. So how did you integrate all those things and to narrow down potential drugs, and how these drugs are working? Like, does it removes the plaques as well as changes the cell fate? And my second question would be like, how does methylation changes the expression pattern in case of a disease condition? And apart from the familial mutations, does any other somatic driver mutations also contribute to this disease?
Okay. I mean, there are 3 questions, and I'll answer each of them. And if I forget, you can remind me. So, I mean, the first question deals with the fact that, how do we reduce all this complex data into actionable consequences? I yesterday pointed out the fact that all this data essentially boils down to, in order to convert them into knowledge, we have to think about models at multiple levels of hierarchy. In my lab, I'm driven by the fact I don't think that I can put everything into a machine and out comes some great result. And that's, in my opinion, a pipe dream. So we use the fact that we have knowledge of mechanisms, knowledge of, various types of phenotypes, knowledge of pathways, so we induce all of those in combination with the data to project them into concrete mechanisms. So data reduction is not just simply you just feed everything into a machine. Data reduction is guided by knowledge-driven approaches. This is the approach we've taken since for the past 30 years in my career. I've just simply followed this logic, and it's worked very well. That's the first answer to your question. Now, the answer to your question with reference to how do you get all these, drugs to really influence or impact these things. So our goal is not to target a drug at a single protein or a single gene, but we want to target it at a whole mechanism, which means the more downstream you target a mechanism, the better off you are because you have less off-target effects. So our targets are always going to be mechanism-driven, where you're going as down the pathway as possible so that you're really not affecting anything else that was happening earlier. And what is your third question? It's a very interesting question.
Third was like, how does any driver mutations?
Oh, yes, very good question. You asked me methylation and driver mutations. Okay. We looked at DNA methylation, and there are some changes, but it is not as dramatic. And I'm talking about not chromatin modification. I'm talking about DNA methylation. It's not so dramatic at the moment in our hands. So we don't think that that's a long-term. It is a really long-term epigenetic change, and we don't see that's a factor. Now, somatic mutations are a different story, which means that what he's talking about, the somatic mutations, is mutations that happen in the lifetime of the disease in the chromatin. There are somatic mutations, there's no question about it, and people are starting to map genome-wide association studies. However, we don't know in late onset Alzheimer's disease, we don't see somatic mutations in the presenilin 1, presenilin 2 APP genes. We don't see that. Maybe there are other genes. I mean, it's a whole background, it's a sea of things. We don't know. So we don't have the answer because we don't have the genome sequence, somatic mutation map, somatic map of all the Alzheimer's patients. When we start getting it, we can do genome-wide association studies. So the jury's out.
So the very last question.
So thank you so much for the wonderful talk. I learned a lot. So my question actually a simple one. In familial AD caused by mutation in PICN1, if I use gene editing to correct mutation, let's say in your iPSC-derived neurons, can we reverse the disease phenotype?
You must have reviewed our paper or something, because one of our reviewers has asked the same question, whether you make the reverse change, you alter the genotype back and see if you're reversing the mutation. So those experiments are underway. Part of the problem in doing that is it turns out that when you do a CRISPR-Cas9 like experiment to do CRISPR-I to change this back into a native mutation, it's toxic to the cells. So we are not able to really sustain that for a long time. So we convince the reviewer, hey, it's a good idea, but that's not the question we are really asking. We are really asking the question of comparative mechanisms, but however we are going to do it. So I just received an NIH grant, excellent score. We don't have the grant yet. We've got a score of 5 percentile, which is going to, we proposed doing exactly that. We want to get isogenic mutations and see if it can restore the disease. So thank you for the good question. So you must have reviewed something.
Thank you so much.
Yeah.
Thank you. I'm sure many of you have questions. We have tea break after this session. Please feel free to talk to Shankar. Thanks so much, Shankar, for the very exciting talk. And please join me in thanking you.
Thank you.
So the next speaker is Dr. Priyanka Bagade. So much. Dr. Priyanka Bagade is a professor in the Computer Science and Engineering Department at IIT Kanpur. Before that, she worked as a software architect in Intel Corporation, US for 5 years. She did her PhD in computer science from Arizona State University. Her current research focuses on the Internet of Things, deep learning, and cyber physical systems. Dr. Bagade's IoT Vision Lab believes that the future is connected and that the power of IoT lies in its ability to create intelligent systems that can communicate with each other, learn from each other, and adapt to changing circumstances. By combining IoT with computer vision, they aim to connect systems that can not only collect and process data, but also make sense of it in real time. Dr. Bagade.
Thank you, Professor Shankar, for the warm introduction. So hello, everyone. So I'm going to talk about various use cases of IoT in healthcare. So you will get snapshots of different use cases. So if you want to know more details, you can ask me questions. But there are so many things, so I thought that I will just show some snapshots. Okay, so before we get started with the IoT use cases or the IoT applications in hardware, first of all, let's try to understand what is IoT. So IoT is basically Internet of Things, and here's the definition that I use in my class as well. So IoT or the Internet of Things are basically small ubiquitous devices which are connected to the internet, and these devices keep on collecting information from the environment. So IoT is not only for the healthcare, but it is also for other applications. So for example, you can think about smart traffic management or smart building management. So anywhere, wherever we want to understand something from our environment, we want to learn the context of the environment, there comes the IoT. Okay. And as it says that it uses small devices. So since these devices are small, you can think about what are the challenges these devices or the systems can face. The first challenge is less power, less memory, less computation power. Okay, so whatever data we collect, we have to ensure that we should be able to process that data or try to understand the context of the environment with minimal computations. Okay. So apart from the temperature sensor, ECG sensor, blood pressure monitoring, here I just enlisted 3 very smart sensors which are being used widely nowadays. So the first IoT sensor or the IoT device is smart banded. Okay, this was invented by University of Glasgow in 2021, and this particular banded, it incorporates temperature sensor, pressure sensor. It measures the skin strain level, and based on that they try to figure out how much the wound underneath the skin has been healed. So patient doesn't need to go to the doctor or the hospital all the time. This sensor directly sends data over Wi-Fi to doctor's smartphone or the computer system, and doctors are able to monitor the patients remotely. Okay, so that even doctors don't need to invest the time. They can simply look at the graph. Okay, this is how the patient is doing, whether the patient needs to if there is a need to change the treatment, whether there is a need to redo the band-aid or any other thing, or change in medications. Everything can be decided remotely. The next sensor or the IoT device is ECG patch. So we know that there are lots of heart patients or cardiovascular diseases are increasing with our newer lifestyle. So doctors and the researchers, they came up with this ECG patch which patients or even normal people can wear all the time. And this patch basically captures the respiratory rate as well as ECG. This is basically 3-lead ECG. It captures that data and send that to the remote doctor all the time. And then again, remote doctors keep on observing the data. Actually, doctors don't look at the ECGs directly. They look at the analyzed data. They just check the pattern, how the ECG is changing, how the PQRS waveforms are changing over the period. Okay. The third one is glucose meter. So you might have heard about continuous glucose monitoring, CGM. It is the new acronym nowadays. So even most of the normal patients, there are diabetic patients. In day-to-day life, they just wear this on their arm to monitor their blood glucose level continuously. So instead of pricking the finger every morning to check your blood glucose level, this basically keeps on checking your blood glucose level, send the data to your smartphone so that even you can look into how your blood glucose level is changing throughout the day. And this also this information also gets sent to the doctor, and then again doctor can look at all these things remotely. So basically with IoT and these health sensors, we can enable patients and doctors to have continuous monitoring and as well as remote inspection of patients' health. So apart from these sensors, camera is also considered as an important IoT sensor, because how do we define a sensor? That the sensor or the device which can understand the context of the environment. And camera is doing the same thing. It is trying to understand the context of the environment with visual aspects. Okay. In this throughout the presentation, we won't we not only will consider the sensors which are looking at the physiological data, but we will consider camera as an IoT sensor as well. So this is a very typical use case of healthcare, that there is a patient who is wearing different wearable IoT devices. These sensors are responsible for collecting physiological data of that patient or even the person. So all of us, we know that we use those smart watches or smart bands, right, which keep on tracking our respiratory rate. Then it tracks our temperature, number of steps we have taken that day, and based on that it calculates how many calories you burn throughout the day. Okay, so even that smart watch or smart band is considered as an IoT device. So we collect all this data, and then that data gets sent to your smartphone, so that you can even take a look at your smartphone app and check that okay, how you are doing throughout the day. So even this IoT sensors are applicable for our day-to-day life monitoring as well. Then smartphones don't have enough memory to store all this data. So eventually, probably nowadays these smart watches, they follow a strategy of sending data weekly, usually every Sunday night, to the cloud. And all this data is stored on the cloud so that we can use different data analysis algorithm to do historical analysis of that patient's behavior or the health conditions throughout many years. Okay, so that data goes to the data center, and doctors are also connected to the data center. So remote doctors can also keep on observing these things. So for normal life, right, when you are just measuring like how many steps you are taking throughout the day, doctors looking at your data won't matter that much. But if you have a critical condition, in that case, this direct connection with the doctor is very helpful. And then in the end, there is a hospital data center as well. So patients in the hospital as well, they can be on these wearable devices, and based on the conditions of the patients, hospitals can actually decide that what kind of drugs are required in this particular areas of the hospital. That's why in the end I call that as a business layer, because these sensor data, they also help hospitals to decide what kind of drugs, what kind of diseases are going in particular area. Okay. So during second COVID wave in India, actually we did analysis with the UP government, where we figured out that what are the hotspots for COVID around Kanpur cities, and then we gave feedback to the UP government, and accordingly UP government actually provided medical drugs in those particular area. And for that we took all the data from the ABHA workers who went to every village, to every person, to collect the data. So that was one of the experiment where I really understood the business part of the IoT system. Okay. So we'll look at one very classic example of the continuous monitoring. So that is IoT enabled teleambulance. So nowadays, like whenever patients are in the ambulance, you can see that those patients, they wear ECGs, or they wear basically something similar to ventilator, like oxygen supply. Their temperature gets measured, their BP gets measured, but everything gets measured and everything stays inside the ambulance. Okay, so during the transit, we are not treating that patient, or whatever treatment is provided, that is provided only at the nurse level, because we cannot have MD doctor in every ambulance. Okay, so we are basically wasting that much time for that patient's treatment. So usually when ambulances are used, whenever there is a heart attack or some brain related surgery or any maternity related or any accident, right? So all these cases are very serious cases which require immediate intervention from the medical professionals. But nowadays, like we cannot provide that. So the proposed system is teleambulance. So all those limitations of the current healthcare system I have just mentioned. So what if we have all the sensors which are used on patient's body inside the ambulance are connected to the internet, and what if we keep on sending this data to the remote doctors sitting in some hospital? So doctors can immediately start the treatment on that patient during that transit, so that we won't even waste that time. Okay, and for that one now, with the increased use of 5G or 5G tower installations, we can send this data quickly to the remote doctors, and during that time even doctors can assess the physiological condition of that patient and decide that which hospital that patient can go to. Okay, so during COVID there were multiple hospitals, but the ventilators were not available. So ambulances were taking these patients to different hospitals, and then the hospitals used to tell that, okay, we don't have any ventilator available. But what if all the hospitals were connected and we had a database, okay, these hospitals have the ventilators available, we could have sent those patients to those particular hospitals directly, instead of wasting time in going from one hospital to the other hospital. Okay, so this is the completely connected emergency healthcare system that can be enabled with IoT devices. And I'm working with UP government and Tata Communications to enable this system. I recently got funding for 5G lab from Indian government to enable this system. So hopefully this should be in picture very soon. Okay. So one thing is patient routing, that I said that, okay, depending on the patient's condition, we can find the nearest hospital, as well as first of all nearest ambulance to pick up the patient, then nearest hospital. Remote patient monitoring, then again that IoT devices, they keep on collecting the data, send that data to the doctor. One interesting aspect in sending the data to the doctor is, here the assumption is we have continuous internet connectivity. However, our vehicle is mobile. Vehicle is not stopped. It's not stationary. In India, when you go from one place to other place, there is no guarantee that you will continuously have communication signal available. Okay, even if I am saying that I got funding for 5G, but I know that 5G is not available. I'm not sure whether 5G even available here right now here or not. I haven't checked my phone, but there are not that many 5G towers deployed yet. Okay, so when we are sending this data, what if the communication channel changes? So what if we go from 5G to 4G to GSM to no signal at all? Can we still work with teleambulance? Okay, so for that one, we developed a machine learning model which can predict the next ECG pick. So we are successful in predicting up to next ECG picks for next 5 minutes. After that, it's very difficult, because if the patient is having heart attack, their ECG changes quite drastically. And if we don't know the patient's ECG beforehand, that means if we don't have the digital twin of the heart of that patient, then this is very difficult. So that's why we are only successful up to calculating ECG picks for next 5 minutes, and we thought that okay, that is sufficient. Another technique that we have developed, that can we send a very compressed data? So if we don't have 5G connection, if we have GSM connection, instead of sending a complete ECG, can we only send particular pics and regenerate that ECG at the doctor's end? And we are completely successful in that. And we are right now testing this in Narayana Hospital, Bangalore. So it's a cardiology hospital, and they are testing the model to ensure that this actually works. And we are also working with them to quickly detect the ventricular arrhythmia, so that data can be used during the heart surgery. So we are working on separating P waves and T waves, particularly for the VT patients, where these P waves and T waves are not visible in ECGs. So can we extract those using some computational analogies? Because even doctors are not able to see it, but they still want to know the frequency. So it's not visually there, but in computationally it's available. So we are working on segregating P waves and T waves from the rest of the ECG. Okay. Another thing is, we want everything to happen in real time. So again, less computation. How can we achieve the best in less computation with less computational power available as well? So we cannot assume that we are doing everything on GPUs. Okay. So Dr. Shankar just gave an awesome talk on the Alzheimer's and the Parkinson's disease. So basically, he looked into the brain cells and how the drugs can perform. My work focuses more on how we can help these patients in their day-to-day life. Okay, so in elderly care monitoring, basically I'm looking at the Parkinson's disease patients. I'm collaborating with the SGPJ hospital, still waiting for the data, but I developed some initial thought process on this. So for the Parkinson's patients, over the period, their gait changes. So gait means their walking style. Okay. And nowadays, like, it is all over the world, this gait is measured by the subject expert. So if you go from one hospital to the other hospital, your analysis might change because it is very subjective, right? And if the same doctor doesn't look at you or doesn't assess your condition for your consecutive visits, that means it can also lead to the errors. So how can we avoid this gait measurement errors and help these patients to get better treatment? So for that, I propose that, okay, we can use our smartphones. In smartphone, we have gyroscope and accelerometer that can measure the way our gait, as well as we can use cameras in the hospital setup and keep on recording all the visits of these patients and use computer vision algorithms to find the minute changes in the gait which the subject expert might not be able to find. Of course, all these algorithms will be trained with the help of the subject expert as well, because since I'm not a medical professional, for me all this data is just numbers. But what are the features that I should look at? Doctors definitely help in all the respects. So even for the ECG extraction, right? For me, it is just a waveform. Means I am not being. I'm still human. I'm not looking at these things as robot. But when I start working on a computer, for me, all these just are numbers, and I'm just trying to extract some waveforms from it, and then doctors decide how to use that data further. Another thing is person tracking and detection. So for the Alzheimer's patient, as Dr. Shankar already mentioned, that they tend to lose their memory. So can we track these people when they are in the elderly care setup or when they are at home, so that we can guide them? Because most of the times these patients are old, they are staying alone at home. So how can we keep track of these patients throughout? So basically we have developed a computer vision algorithm where even though there is no overlapping area between two cameras, right? So for example, we can have camera in the living room, we can have camera outside the home, but we cannot have camera in the restrooms or even in the bedrooms, right? So can we still detect that person even if that person goes from one area to another area with different clothes? So we proved that okay, it is possible using computer vision algorithms. And another thing that we are working on, what if we have non-vision sensor in one room, that means motion sensor, and we have camera in another room? Can we still detect the same person walked from one room to the other room? Okay, so another important aspect in security in IoT devices is security. So I showed you the continuous glucose monitor. So this might be a shock for all of you, but none of the currently available continuous glucose monitors use any of the security protocol. They simply measure your glucose level and they send the glucose level, the value as it is in plain text, to your smartphone through Bluetooth channel. So any attacker or hacker can actually modify that value right away. Even I can do it right away within 2 minutes. And then your smartphone will start showcasing random values. Your doctors will start giving you some random medications because doctors just have that data. Okay, so it is scary. So that's why it's very important for us to look at the security as well. Yes. Another thing is, as I just alerted all of you regarding how hackers can play with your health data. So digital forensics of medical devices is very important. Currently, I'm collaborating with GE Healthcare, which develops different medical devices. So they are getting different legal cases on their company for these hackers basically attack their medical devices, and patients are saying that the patient's family is saying that patients did not die because of the physiological changes that happened in the patient, but the patient died because of the cyber attack, and that attack has actually changed some of the parameters on the medical device. So for example, ventilator is responsible for providing a certain amount of oxygen to patient's lungs, but what if the ventilator gets attacked? Then the input oxygen level will change. It might go to 0, or it might be too large to be handled by that patient's lungs. Then the patient will die. Right? And these are true cases. So I'm working with GE Healthcare to analyze that, okay, how these attacks happen. Can we detect these attacks when there is remote communication happening with these medical devices? Because all these devices are connected to the internet, because that's how doctors can monitor them remotely. So for all ICU patients, can we monitor all the data traffic going through these devices? Another thing is, whenever doctors actually look at the DICOM images, the CT scan images, MRI images, the DICOM has a feature that it has the empty space in the image, which allows doctors to open that image on their smartphone, on their tablet, on their Linux system, on their Mac, on their Windows system. But this feature also has a drawback, because it allows these different formats of the image. It has lots of empty space. So that's where the attacker puts the malware. As soon as the doctor opens the image, it inserts the malware into doctor's system and it just changes the image. So then doctors start taking wrong decisions or might take wrong decisions. So I'm helping GE Healthcare to look at all these attacks and also look at how to retrieve their medical device from these attacks, because these devices, they are very expensive. And once the malware goes into any medical device, or for that matter any device, it's very difficult to get rid of that malware. You have basically need to scrap that system completely, but we cannot afford to scrap medical devices. Okay, so we are working on how to look at these malware, how do they work, then generate the evidence for the codes to show that, okay, these are the affected areas of the medical device and how this is how we have retrieved the system back. I already talked about the intrusion detection. So we are using machine learning algorithm to basically analyze all the data traffic. Again, also analyze the memory of these computer systems to ensure that, okay, when this malware process ran on that particular device, what are the changes that happened on that system's memory, and what was the memory before the attack happened, and what was the memory content after the attack has happened. Another issue that we are actually facing here, all these devices, they have very small memory. So even if we decide to keep on storing the snapshots of the memory after every 1 second, right, it's not possible. So we have to smartly decide when to take that snapshot. So for that we are developing another machine learning algorithm. So this is basically a nest of machine learning algorithms to do different tasks. So smart hospitals, there everything is connected. Another interesting topic is remote surgery. So we know that, okay, lots of medical schools are there, new doctors are coming up, but when you actually visit the hospitals, you realize that, yes, 2 minutes? Okay, I will finish very quickly. So you realize that, okay, there are experts who are about to retire and new doctors are still learning from these experts. Okay, so some of the decisions these experts take within a second. Means I have seen that in SGPJ hospital in Lucknow, but the new doctors, I don't want to scare any of you, but they take around 6 to 7 hours. So we are looking into how this process can be accelerated. So we are looking into medical image analysis as well. So in remote surgery, these doctors can actually advise their colleagues who are new into surgeries remotely as well, irrespective of where they are sitting. So we can have cameras installed in operation theaters, OTs, and remote doctors can keep on looking at what are the things that are happening. And then we can use robots as well. So robots contain different IoT sensors so that the remote doctors can keep on looking at the robots. Another thing is during, this is the last point that I will cover. So during surgeries, around 300 to 600 small different tools get used even for the small surgeries. About 10% of the surgery implications that happen in that one, 10% are related to that some of the tools, they still remain in person's body. So I'm working with SGPJ hospital to keep track of all these tools that are getting used in the surgeries, so that we get to know that whether any tool remained in patient's body. So that is, I guess that's it. This is basically using drones to collect pathology samples from different places in villages or in remote areas and send that to the pathology labs so that the testing can be done, and then again remote patient treatment that can happen easily. So I guess that's it from me. Thank you.
Thank you, Dr. Bagade, for providing a future of healthcare, glimpse of healthcare industry. You have There are a few questions. Shankar first.
Dr. Bagade, thank you so much. It's amazing and it's really very, very important work. So my question to you is the following. Both in the case of where you have failure of internet, 5G, 4G effects, as well as in terms of security information, the current thinking in US is that longitudinal measurements and modeling of longitudinal data can be used as an input feature for detecting, for example, errors, for detecting, intrusions, because just like Fourier averaging, you don't average over all those things in an intrusion. So are your approaches with GE, for example, are they using longitudinal data? And the follow-up question to that is, AI ML is not very good at dealing with longitudinal measurements because they treat time as another lump parameter. So are you developing methods to look at time as a unique dimension in terms of learning algorithms to bring feature evolution across time as a factor? Are you working on those things? Just curious to know.
That's a very good question. So we are looking at the time-based data right now because the attacks that happen on the medical devices, the attack doesn't happen right away. It's a slow modification in the data. So even attackers are actually very smart. They don't want you to let the patient or any intrusion detection system know that the changes are happening. So for example, if your blood glucose level is right now suppose 120, this is just an example, okay, it won't suddenly go to 260, it will go to 121. Slowly, day by day, they keep on making changes, so that you have to continuously observe how the changes are happening, and you have to continuously predict that, okay, if the person is having this kind of food and based on the history, whether this should be the next glucose level or not. So you do prediction and you do detection. So these two algorithms, they run in parallel, and you keep on checking and do cross verification. So this is for the glucose monitor. But the medical devices that I'm working with GE, basically we are looking at the memory footprint right now, because that is the most troublesome area, and there are no tools available, and nobody in the research, according to them, like no researcher actually works in that area because it's very difficult. So we are trying to explore that at every second how the memory is changing, and since we can't store it, we are training the models, and we are also storing certain, instead of just storing the models, but we are also storing some memory parameters somewhere. So basically we are right now we are doing it in one of the local system, because G doesn't want to send anything to cloud. Did I answer your question? Yes, we are not considering right now because they are mainly they want me to look at the time series data only, but that's a very good thing that I will talk to them.
I think there was somebody who raised hand. Yeah, please go ahead.
Priyanka, you gave a wonderful talk and it was very interesting to see the things you are doing. So my question is, the ambulance you were showing, and you were showing that it has a number 108 and it has the connectivity to the professional. But the real scenario in India, especially in UP, like there is 1:22,000. Like a patient doctor ratio is so high. Even patients who go to hospitals, they are not attended by the doctors when they are actually should be attended in their critical time. So is there anything in your ambulance which can help the professional which is there with them to get some primary treatment till they are connected to the professional?
In ambulances, nurses are there. So even though they are not experts, they can provide the initial treatment. And the model that I am talking about for tele-ambulance, there will be doctors sitting who won't be directly treating the patients, means they won't be connected to the hospital, means they won't be waiting for the patient and start treating the patient. These doctors will be there only for remotely monitoring the patients. So patient will keep on getting the treatment during the transit. So once that patient reaches the hospital and start getting the treatment in the hospital, then this remote doctor will get disconnected from that, and he or she will start communicating with other doctors.
So there will be a different set of doctors, especially for these ambulances. Okay, thank you.
SGPJ hospital is basically for creating MD doctors, right? So they train MD doctors. It's a medical school, right? So in their 3 hours of education there, every day they have to sit in front of these remote systems for 6 hours. It's mandatory for them, and they remotely treat the patients from villages. So these patients come to the kiosk where these cameras and different health sensors are installed. So the nurses which are there in those villages, they take all the data from these patients, and then these remote doctors, they look at the patients through video conferencing. Okay. And there are around 200 such kiosks installed, at least in UP, that I know because I visit SGPJ quite often. So I have seen that these doctors, they have to do this duty every 6 hours a day. It's compulsory for them.
Okay, thank you.
Yeah.
Professor Katti.
Very interesting talk. I just wanted to know, so many of the things that you mentioned, they talk about taking data from one location to another location. Now, outside any hacking or malware, what are the parameters that determine the quality of the data? Because treatment is based on quality. So that the quality of the data that is transported is very high quality. That means at the source and at the end where it is received. What are the parameters that can affect the quality of the data?
Yes. So we can look at these from the 2 points. First is just the data where numbers are there. So and another thing is medical images. So when we look at just the data, right? So ECG, even though it's an image, but it is actually a combination of PQRST waveform. We can send ECG as the image. If we can send that image as it is in 4K format, then doctors will receive that in the 4K format and they will be able to see. If we have 5G connection available, we can do that very easily and it can happen in real time. Okay. But what if we don't have that connectivity, right? Then we are only detecting the PQRST peaks and then we are redrawing that at the doctor's end. So that might not be accurate because PQRST, do I have that wave? Just a minute. Okay. So you can see here, right? These are the threshold values. If we don't have good connectivity, we are only sending these threshold values. That means we are only sending numbers. We are not sending the complete wave. Then we are regenerating that at the doctor's end using some model. Thank you. Then we are generating that at the doctor's end with some model. But there can be some waveform disruption here, and that might lead to some new information. Right? So that in that case, we can say that we are not sending good quality data because we don't have good connectivity. If we send the complete data, then that's a different case. Same thing happened with medical images. So whether they are X-ray images or CT scan images, if you reduce the resolution rate, so it is, imagine that when you had those small phones whose camera was not good, and the current phones where we can take very good pictures, that's the difference in quality. And then based on what kind of quality data or what is the quality that the doctors are receiving in the end, their treatment or diagnosis might change. So that's why for ventricular tachycardia, we are trying to separate these waves. So instead of sending, if the connectivity is not good, if we can just take the T waves out and send those T waves to the medical professionals instead of sending all the PQRST waveform, then that will help the doctors to take better decisions.
So the question I also had was that does it only depend on connectivity, the quality of the data at the recipient's end, or does it depend on anything else?
Yes. So connectivity is one part. Another thing is the sensor resolutions as well. Again, the camera, right? what is the resolution for that camera. That is one thing. Even in the ECG, if you have those 12 lead ECGs, right, you get excellent ECG out. But the patches, that doesn't provide you good ECG out. So it also depends on what kind of sensors you are using.
Okay, thank you.
Yeah.
Thank you, Dr. Bagade. I hope some of these things will be realized as part of the MFCEM.
Yes.
Thanks a lot.
Thank you.
Yeah. Thank you. Please join me in thanking her. So now we have lightning talks by students. The first student speaker is Ajita Shri. No. It's not the same student. It's a different student.
Do you want to?
Good morning, everybody. My name is Ajita. I'm a PhD student in Dr. Hamim Zafar's lab, Department of Computer Science and Engineering. Today, I'll be giving talk on SCREMA for Atlas level integration of single cell dataset using deep generative model paired with adversarial classifier. This is a joint work with another PhD student, Krishna Pawan. So I'll start by giving a brief overview of single cell data. So it is through single cell sequencing technique that we are able to measure the gene expression at a cellular level in any given tissue. And hence, we are able to resolve the heterogeneity in any complex system. So single cell data looks like cells cross gene, where each entry defines the expression of that gene in that cell. But there are many challenges in single cell data because it's available from heterogeneous sources, sometimes from multiple labs, sometimes from different sequencing protocols, different donors and tissue samples, because of which there is presence of batch effects if we visually see the data. Here we can see in the diagram that the cell types are, so for example, cell type macrophages is spread across the whole plane because it is contributed from different batches. And integration is necessary so that we can have a nice representation, as we can see in the middle diagram, where similar cell types have come together in a cluster and the corresponding batches have been mixed. It's a primary and fundamental step because after integration, we can do any downstream analysis. So we present our method, SCREMA, a framework using which we can do different kinds of integration. It's a deep generative model that do adversarial training, and it has multiple components using which we can do unsupervised integration, where we do not use any cell label information. When we are utilizing cell type annotation in the data, then we do supervised integration and semi-supervised integration. There are two goals of integration. First is bioconservation, and the second goal is of batch correction, as explained in the last panel. So we have demonstrated the performance of our method on a variety of challenges. First is the lung data, immune data, heart cell data, cross-species integration, that is human and mouse integration, pancreas data, and monkey retina data. I will be briefly explaining human-mouse results. So on the left-hand side, we can see the unintegrated data, and on the right is the output of SCREMA, where we see the batches have been integrated well. And in the last panel, we quantitatively see how the method is performing better compared to state-of-the-art method in terms of bioconservation. Second plot says about batch correction, and the last plot talks about the combined composite score, taking into account the holistic performance in both of these metrics. So here we are seeing, so we did this study to see how well our method is able to find some biological insights in the data. So we observed dendritic cell population. We found some study by Villani et al., where we found out that dendritic cell population has multiple subcell types, DC1, 2, 3, 4, 5, 6. And we did this analysis in our method. We did the subclustering and found out that our method is able to capture these subcell types pretty well. Whereas the second best method called SCVI, these subpopulations were mixing with different cell types, and which is not ideal. We did a further comparison with another best performing method called Harmony, and here we observed that it is not able to find out DC1, DC5, and some subcell types well. Lastly, here I present a million cell data set called Healthy Heart. It has 0.5 million cells and large number of batches, 147. And here also we see that our method is able to integrate the data very well, as we can see on the left. And on the right, it's the quantitative comparison. In both unsupervised and supervised category, SC-Dreamer is performing the best. Lastly, we manually removed some of the cell annotations from the data, 20% cell label, missing cell label, and 50%, to see if the performance is remaining best in that category or not. And we see that other methods' performance have dropped, while SC-Dreamer is able to maintain that good performance. In conclusion, I would like to say that SC-Dreamer is a framework which is very scalable with respect to number of cells, batches, and it can do a variety of different integration tasks. And it is outperforming other methods by a large margin. And the link for the bioarchiving tutorial is available here. In acknowledgement, I'd like to thank my advisor, Dr. Hamim Zafar, IIT Kanpur, Department of Computer Science, Mehta Family Center of Medicine for giving the opportunity, and Cosmic Lab members. Thank you, everyone.
Thank you, Ajita Sri. I mean, if there is one question, I will allow, but we are running. Okay. So the next speaker is Aditi Ladda.
Good morning, everyone. Today I will be presenting part of my PhD work stating as sweet transporter and cadal receptor, evolutionary and functional studies of the sequentially diverse membrane protein with the similar structure fold. So, as we know, in a eukaryotic cell, there are many processes which are important. Two of these processes are protein trafficking and sugar transportation in plant. So, in case of protein trafficking, as we know that after the synthesis of protein, they will get transported to the Golgi apparatus for their modification. But in order to do so, some of the ER resident protein, which helpful in the protein proper folding, also get transported from ER to the Golgi apparatus. So in order to bring them back, there is a receptor known as cadal receptor help to recognize the C terminus of the ER resident protein and bring them back from the Golgi apparatus to the ER membrane. So, and one of the other process in plants is the transportation of water and mineral from the roots to the shoot, and other is the transportation of sugar from the leaves to all the other part of the plant. So, one of the protein which is important in the transportation of sugar is the sweet transporter. So, these transporter help in the efflux of the sugar molecule from the parenchyma cell to the apoplastic space, and then after they will be uptaken by other transporter to transport in the whole plant. So in order to know more about these protein, researchers have crystallized the two protein structure. First is the cadal receptor, which is recently crystallized in 2019, and second is the sweet transporter. So by looking into these transporter, we can see that both of these transporter are adopting the same structure fold, that they are having the 7 transmembrane region, and these 7 transmembrane region are arranged in 2 triple helix bundle, which is connected by a linker helix, which is TM4 triple helix, which is the linker helix number 4. So when we superimpose these two structure, we found out that the RMSD value is quite low. RMSD value is quite low, which is 1.05 angstrom. So we were curious that how two functionally very diverse protein are adopting the same structure fold. So in order to do so, we want to first know about all the residues which are conserved between firstly in the Cadle and in the suite, then we want to compare them. For that, we develop a methodology where we extracted the data from the UniProt, and then we clean the data where we remove all the sequences having more than 98% sequence identity. Then we remove all the false positive result from our data and did a structure based sequence alignment. From that alignment, we identify all the residue which are conserved in Cadle receptor and did a phylogenetic analysis. So all the important findings from this pipeline were compiled in the form of a repository known as DB Cadle receptors. So similar work has been used previously in our lab to develop another repository for the suite transporter where the variation in the sequences are present. So next we want to compare these two sequences from these two proteins and found out what are the residue which are important in both of these protein and conserved across the protein. So this is the representative logo for the two protein sequences across all the kingdom, where we can see that this is the triple helix bundle 1, triple helix bundle 2 in case of suite transporter, and here the Cadle receptor logo is presented. So from our analysis, next we did is a phylogenetic analysis for the Cadle receptor. So we found out that Cadle receptor are classified in the three major clades. Clade 1 is a fungi, which is represented in the red color here, having 608 members. Clade 2 is plantae, having 429 members, and the clade 3 is metazoans, having the 658 members. So these three clades are having some similarity but also some differences. So some of the differences we can see in the right side, that some positions are different, and we also did identity and similarity amongst the clade and between the clade. Next, when we compare the residues between the suite and Cadle receptor, we came to know that there are 20 positions which are conserved. Their chemical nature was conserved. Like if there is hydrophobic residue, then it is conserved. But we also find out that the most conservation is in the helix-helix interface residue. So as we know that helix-helix interfacing residue are important to maintain the integrity of the protein. Next, in this movie, we can see that the Cadle receptor and suite transporter structures are superimposed, and all the conserved residue, 20 conserved residues, are present here as are color-coded aliphatic hydrophobic, weakly polar, aromatic, polar, and glycine and proline residue which are conserved. So from this study, we conclude that there are helix-helix residues which are very conserved and which suggest that these two proteins are, this can be one of the reason how these proteins are having functional differences but still are structurally adopting the same fold. So currently I am using molecular dynamic simulation to determine recognition and transport mechanism in the Cadle receptor, and also I am using umbrella sampling to determine the selectivity mechanism in the suite transporter. Last but not the least, I would like to thank my guide Dr. Shankar and also my lab member and HPC facility and IIT Kanpur for providing the assistantship for the fellowship. Thank you.
So the next student speaker is Dr. Mr. Amal Jude Ashwin.
Good morning everyone, and welcome to my talk on the topic 1-minute cognitive physiology signals are able to predict the treatment outcome of depression as early by 2 weeks. So the research work is done by myself, the one with the secular name Amal Jude Ashwin, under the guidance of Dr. Priyanka Bagade, with the support of my colleague Mr. Hari Kumar Tiwari at the Translational Neuroscience Lab, Department of Cognitive Science, IIT Kanpur. We are collaborating on this project with Dr. Aloj Bajpai and Dr. Nandini Priyanka Balasubramaniam. Suppose we have a person who's feeling really low and hopeless and is willing to seek professional help. When the person visits the clinician diagnoses the participant based on something called PHQ-9, which is a 9-question questionnaire that covers the symptoms of depression. So we have symptoms of depression like hopelessness or thoughts of suicide, the differences in appetite or sleep pattern. So the 9 questions score from 0 to 3 each, where 0 corresponds to healthy and 3 corresponds to depressed participant. So therefore it's a 0 to 27 questionnaire. And based on the answers given, the person is diagnosed with depression and is recommended medication, which is the first line of defense. And the clinician asks the person to follow up after 1 month. But the catch over here is antidepressants work only for 40 to 50% of the patients who are prescribed medication. So what about the remaining 50 or 60%? 1 month is definitely a long time to wait to see if the medication is working, because depressed participants tend to go to extreme measures. Therefore, can we reliably early predict the treatment outcome? So we have the baseline PHQ-9 from the clinician and the follow-up after 30 days. We propose to use EEG at a follow-up visit at 7 to 14 days. So using the change in EEG values, can we reliably predict the change in PHQ-9 scores as well? To translate it better, can we reliably predict change in mental health using the change in neurodynamics across visit 1 and visit 2? That is as early as 7 to 14 days. So in order to do that, we collect the EEG signals while the participant is performing very simple cognitive tasks like eyes open, eyes closed task, the breathe in, breathe out task, and an MMSE, which is a questionnaire that tests your cognitive ability, and finally varying frequency photic task where a light blinks at different frequency. It's a cue attention task. So we extract EEG features, and along with the EEG features, we also feed in the demographics of the participant, and the individual question can answer to the questionnaires that they are performing. So we collect the demographics and questionnaires during the first visit itself. So we feed it to a predictive model. In our case, it's a logistic regression model, and predict if there is positive change in mental health or no change in mental health. So we collect the data, we pre-process the data, and we extract EEG features such as a functional connectivity measure, which is called coherence, and various band powers. So we scrutinize the EEG features based on 3 criteria. So criteria 1 tells us if that particular EEG feature can distinguish healthy and depressed participant. Criteria 2 tells us if that particular feature is sensitive to the change in mental health. And criteria 3 tells us if that sensitivity to change in mental health, is it reliable or not? So based on these 3 criteria, we filter the EEG data, and we add the demographics and the questionnaires, and we develop a predictive model. So we perform sequential feature selection to further scrutinize the features, and we end up with a bunch of 14 features which are a part of demographics, the EEG features, as well as from the questionnaires. So the coefficients that we see over here are the logistic regression coefficient values for the corresponding predictors. So we were able to predict with an accuracy of 95%, that is 46 out of 48 participants. We were able to predict if they are showing positive change in mental health or no change in mental health. So we performed stratified 5-fold cross validation to get the following metrics. And when we plot the sum of weighted activation, which is taking each predictor over here and multiplying it with the corresponding coefficient value and summing it up, we can see a clear difference between the participants who are showing no change in mental health and positive change in mental health. When we change the y-axis of this plot even better, when we plot the sum of weighted activation against the PHQ-9 difference, we can observe that for participants who are showing positive change in mental health, it follows a linear trend. So therefore, we can also predict if they are responders or they are remitting to the treatment that they have been given as early as 2 weeks. So in order to further explain the results, when we go to a clinical setting, we can't just go with a model. We have to explain why for that particular patient they are getting predicted as whether they are showing positive change or no change. So in order to do that as a future direction, we have to explain the neural features that we are extracting and also provide subjective analysis. To validate the results of the model that we have got, we are collecting further patient data and we are hoping to scale it to other treatments for depression like the repetitive transcranial magnetic stimulation or even cognitive behavioral therapy. Or in the future, hopefully the same methodology, if it's foolproof, can be extended to other mental disorders as well. So to conclude with, mental health matters. Thank you.
Thank you, Amal. The last speaker is a BTech student, Kaushik Raj.
So, hello everyone. My name is Kaushik Raj Nada and I am a fourth year undergraduate student at IIT K, pursuing a dual degree MS in Statistics and Data Science and a BTech in Biological Sciences and Bioengineering. And now I will discuss my work on pattern recognition for electromyography signal, which I did as a summer internship at Life and Limb Private Limited. So, first, what is a prosthetic device? So it is basically an artificial device which is a functional replacement for a missing or an amputated limb. And this diagram shows the basic components of a prosthetic limb. So it consists of three components. One is the terminal device, which contains the microcontroller, the motors, and the battery. And one is the shell, which provides the structural support for the device. And the third is the socket, which is the main contact with the skin and contains the surface electromyography sensors. And basically how it works is the sensors record the electromyography signals from the skin of our hand, and based on those signals the microcontroller decides which motor to activate and controls the hand. So the current problem in this is that basically there is a button for each, there are presets for each activities which we can do with our with the prosthetic device, and using that button on the terminal device we can switch between different modes, and there is a certain threshold after which the EMD signal detects and the certain motors are activated. But in this model the problem is that every time the user needs to switch to different activities, he needs to click the button using his other hand, so it is very inconvenient for him. So what I have proposed is using a machine learning model which can automatically detect which grip patterns the microcontroller needs to set and which motors to be controlled for each finger movements. And before workflow I will talk about how I collected the data. So basically I created an automated UI which contains a timer, and after each 5 seconds basically the gestures are alternated between a rest gesture and a non-rest gesture. So for each for this project I chose 12 different non-rest gestures and collected data only for those gestures. So after every 5 seconds, in the first 5 seconds the patient had to perform a rest position using his hand. In the next 5 seconds he had to perform any non-rest gesture which was controlled, and in this way data of around 2 minutes was collected for each gesture. So this is how I made. And so the overall workflow, basically I'll also formulate the problem. Basically the problem is that the input is a time series EMG signal, and the output is basically the classes of gestures which we need to predict. So it is a multi-class classification problem. So in this workflow, we have the time series EMG signal as the input, 2 channel, and we perform segmentation on those signal, which is basically a sliding window of around 25 to 100 data points, whichever we choose. And this window is this window is used to perform segmentation on the EMG signal, and for each windows we perform feature extraction, and the feature vector is associated with that window, and also a label is associated with that window, and the label is chosen by choosing whichever is the majority label in the 50 to 100 data points. And based on these segmented windows, we perform multi-class classification. We train the machine learning model on these 12 non-rest gestures, and we get the final prediction. So first I tried using traditional machine learning models. So these were the results which I got. These are pretty good for traditional machine learning models. These accuracies, which are almost all the models give about 90% accuracy. Although the problem here was that I had used a window size of around 100, which corresponds to around 1 second. So which means that there was a lot of delay between the gesture and the prediction. So to optimize this problem, I tried to reduce the window size and perform the prediction, but the accuracy dropped drastically to around 60 to 50%. So these models are not useful in the practical scenario. So I went on to use deep learning for this task. So I chose 2 different types of architecture. One is the convolutional neural networks, and one is the recurrent neural networks. So convolutional neural networks are known mostly for their ability to capture spatial variation in the data, and the RNNs are known to capture sequential variation in the data. So I decided to choose these. And if you see in both these architectures, I have a common thing. In the first layer, basically I am doing a projection such that the lower 2-dimensional data, 2-dimensional EMG data is projected into a higher dimension. So this, I think, acts as a decomposition operation for the signal, and it captures more patterns in the data. So after that, it is mostly a simple CNN or RNN. So using these models, the results which I got were really good. And as you can see on the right, that even after reducing the window size to 250 millisecond, I was able to get very good accuracy of around 99%. So, and also I would like to mention that I had implemented attention mechanism for this. Like in this diagram, I have not shown the attention mechanism. So attention mechanism is basically a weighted sum of all the hidden states of the RNN, and instead of just using the last hidden state, so it gives a better prediction. Also, I have tried. Also, the attention mechanism gives us a good visualization of how the model is trying to learn. So we can see using this attention heat map that where the model attends mostly while learning the data. So yeah, and I would also like to talk about the challenges in this. Like currently, these models were tested using done on the computer. So deploying it on a microcontroller can be a very difficult task, as it will require more compute resources. And also, like for deployment, what we can do is we can send the data collect the data on the microcontroller and send it to a cloud computing system, which will train the model, and then it will we can download the model from the central system and then deploy it on the microcontroller. And modern developments in TensorFlow and PyTorch are such that after we compile the model, they have very good optimizations in such a way that the inference time can be reduced a lot. So these are the future directions. And thank you all.
Thank you, Kaushik. That's a very interesting area. So that brings the end of the digital medicine session. And I would like to thank all the speakers. I mean, very interesting session. And so please join me in thanking all Professor Shankar Subramaniam, Professor Priyanka, and then all 4 student speakers. Thank you. So I have one small announcement. We are running little late. The next is tea break. It is supposed to be from 11:30 to 12, but we are going to have a very short tea break. So we'll be back at 12:15 to interact with the international speakers. Thank you.